physiology, and thinking about how microbes get enough ATP to stay alive, and they need certain carbon sources for that, or inorganic substances for that. Some microbes use oxygen, some don't. But when you think about how they're doing it, I like to think of it as kind of a game. Like, what game do they have to play to get their goals accomplished and win the game? Winning the game would be get enough ATP so that you can have that chemical energy necessary to survive. And so these are the three big picture ideas. It's either through oxidative phosphorylation. So when I say oxidative, am I thinking solely of oxygen? Head shake. No, no, no. Okay, so in this class, it could be oxygen that I'm talking about. But really, when you think of the phrase oxidative phosphorylation, if you just back off oxygen and think about oxidative means stripping electrons off of something. So think about, okay, if you take electrons off of something, you're going to pass it through an electron transport chain. And through that process, so they created a game, so to speak, through passing electrons from donor to acceptor. It creates a situation where they can get protons pumped across the membrane. And that's what they're trying to set up, because then they have that proton gradient, which is potential energy to do something. And for this topic, we're thinking about making ATP. So oxidative phosphorylation, you're stripping electrons, sending them through a chain, donor to acceptor, all the way down to some terminal electron acceptor. It might be oxygen, but maybe not. But oxidative phosphorylation, the phosphorylation idea is right here, where you have ADP and inorganic phosphate reacting. Okay, so, whoops. So really the goal, oh boy. I know. So the goal here, this electron transfer chain process, they're trying to create a situation to get enough potential energy to make ATP. They don't care that the end product is water. That doesn't matter to them really. They just kind of excrete that and don't worry about it. But in that process, they set up the situation where they get a proton gradient. And if you get a proton gradient across a membrane, you should immediately start thinking about, well, that's a great way to make ATP. Make sense? Okay, so oxidative phosphorylation in this class, just think about electrons getting sent down a chain. There will be some terminal electron acceptor that will stop the process, be the final acceptor, but it doesn't have to be oxygen. So we looked at the other idea where maybe instead of oxygen, it's nitrate. So here, this is still oxidative phosphorylation. Both of these examples are oxidative phosphorylation. Yes, on the left, you do have oxygen as the terminal electron acceptor, but on the right, you have nitrate. But it's still oxidative phosphorylation. So this is a very common strategy. Lots of microbes will do this electron transport chain process. Your mitochondria, which were once free-living bacteria, they're doing that in you right now. And really the goal here is to get that proton gradient and get that ATP. Questions on oxidative phosphorylation? Okay. So what we just went through is pretty much what I'm asking you to learn. You do not have to know all the players in the game. Like, what are all the donors and acceptors? Okay, so don't get bogged down in the details. That's not what this class is focusing on. You need to know a little bit of physiology so you can better appreciate why microbes are showing up in certain habitats and not in others. And also to impress on you how incredibly versatile they are in terms of how they get their energy needs met. Then we looked at substrate level phosphorylation. This is another big concept. You can do this. You are hopefully doing this right now with glycolysis in your cells. And with this idea, substrate level. So one of the substrates in the reaction pathway is phosphorylated. And at that substrate level, you will get a transfer of phosphate from the substrate onto ADP, and that will get you ATP. So you have substrate phosphorylation going on right here and right here. So 1,3-bis phosphoglycerate donates off a phosphate. That substrate donates off a phosphate to the ADP, and then phosphoenolpyruvate is phosphorylated, so that phosphate gets transferred. Lots of options if you're a microbe to do substrate level phosphorylation. So there's, and really what we're thinking about here is, you know, if you're wondering, well, how does the microbe decide, so to speak, which one to run? It's all based on their environment, their habitat, what's available to them. Do they have a lot of glucose? Then they'll run glycolysis. If not, if there's some other habitat where that's in short supply, they have other options. So basically, I like to think of it as microbes walk into a pantry and go, well, what do I got to cook with today? And they have to think about what's available and bring that in and start running the appropriate reaction pathway. So they have lots of options, and you can force the situation if you want, like we talked about last week with ethanol production. You know, you are manipulating their habitat very precisely, and you are kind of pigeonholing them into, I only want you to make ethanol. And so you can do that in the lab. You can manipulate by how you feed your microbes to turn on certain reaction pathways that you want. So when I'm looking at my Streptomyces bacteria and I want them to produce antibiotics, I have a certain recipe I'll make and grow them in so that it's forcing them to turn on that pathway. So microbes can be manipulated that way experimentally or in industrial microbiology, as long as you get to know your microbe a little bit and figure out what they're capable of physiologically, then you can very much influence that and manipulate it. Okay, so substrate level, questions on substrate level? We talked about anaerobic too, you know, with that, when going back to this. So these are both oxidative phosphorylation, but this right one also is anaerobic. Okay, so anaerobic metabolism. So when you hear that phrase, they're trying to get you to think about, yes, you have an electron transport chain process, but you just don't have oxygen. Okay. Then let me get back to where we left off. So chemolithotrophy, we can't do this. So some microbes are very good at this. They're able to take inorganic compounds around them and strip electrons off of them. And as they're doing that, they'll send it through an electron transport chain. So instead of like what you're doing right now, you're taking glucose and running glycolysis, they can strip electrons. They don't do glucose, they'll use something else. And so we went through the idea that there's lots of options that are out there, and a lot more because humans of human activity too, providing some inorganics for them to use. And then we said, many of these microbes, they take carbon dioxide and make sugars from that. So that is, you know, thinking to how plants get their sugars made. But they are not doing, you know, chemolithotrophs aren't doing photosynthesis, but they're still able to take in CO2 and make sugars, similar to how plants do it. And so from that perspective, really, chemolithotrophs, we consider them primary producers like plants and algae and cyanobacteria that are able to make sugars. So chemolithotrophs are also primary producers, but they never get mentioned when you're talking about primary producers. Because people who teach that usually aren't microbiologists. So this is, I think, close to where we left off, looking at the idea that there's options. And they're using hydrogen gas as an electron donor, iron, ammonia, various nitrogen compounds. CH4 is methane, sulfur. So they have lots of options. It depends on where they live and what they've adapted to. And you can see a lot of them choose oxygen as a terminal electron acceptor. That's what's being shown on that last column. And so if you are a savvy microbe, you'll pick oxygen because you get the best amount of ATP from that. Oxygen's so good at accepting electrons, it creates a situation where you get some good potential energy to make ATP. And then we looked at hydrogenotrophy. And again, hydrogen is kind of the opposite of oxygen. Hydrogen's like super good at donating electrons. I always think of it in my head as Tinkerbell with her little wand, and she's going around and doing the pixie dust. And she's like, here, a little pixie dust for you and for you and for you. And that's what I think of. This hydrogen gas is like, yeah, I'll give off electrons. I like to share. Right. So they're just really, really good. So when the fancy way to say that is they have sufficient reducing potential, it just means that they have really good energy potential to go ahead and donate off electrons. And so that's what they're trying to show here, where you have hydrogen gas donating off electrons to oxygen. And again, this is all using an electron transport chain process. So all of these reactions that you're looking at, what they're doing is something similar to this, where they're taking hydrogen gas, stripping electrons off of it and sending it through an electron transport chain process, donor to acceptor, until you reach a final or terminal electron acceptor. In this case, it's oxygen, because this microbe is smart and can use oxygen. But the goal here is not to make water, it's to phosphorylate your ADP and get your ATP. And then you see down here, they are grabbing CO2 from the atmosphere and making sugars off of that. So they're still organic beings, even though they're stripping electrons off of inorganic molecules. So they're rock eaters. Lithotroph are rock eaters. And again, it depends on the microbe you're thinking of and their habitats, what will be their electron source. But again, if you are a savvy microbe, if there's hydrogen gas around, you will grab that hands down, because that creates a situation where you get a lot of ATP. Questions on lithotrophy? The last idea, big major concept, so going back to this, is photophosphorylation. So photo, light, phosphorylation, that's referring to ADP getting phosphorylated. So the use of light, those of you who have taken some kind of plant class should be familiar with that. This is a big picture idea of photosynthesis inside a chloroplast in a plant. Remember, chloroplasts were once free living bacteria. So here you're looking at the idea of light energy striking pigments. So you're looking at chlorophyll, which we often abbreviate CHL. So chlorophyll is embedded in those membranes inside the chloroplast. And chlorophyll is really good at absorbing red and blue light energy from the sun. And that is what the plant is using. That's the energy source the plant is using to get electrons stripped off of water. Water doesn't like to give up its electrons. Water is a hog and likes to keep, it's very selfish, wants to keep its electrons. So to strip electrons off water, you need a big burst of light energy to do that. And so that's what's going on there. And they're not showing the electron flow in this image. I'll show you another picture. But basically, if the plant is successfully stripping electrons off of water, one of the products in that process is oxygen. So that's why we have plants and cyanobacteria to thank for generating oxygen for us over many, many millions of years. But you can see that when you are having this process running, those electrons get stripped off water, get sent through an electron transport chain, and you see right here the payoff. What's the payoff? It's setting up a situation for ATP to get produced, and then this other molecule, NADPH. NADPH is what we call a reducing molecule. So NADPH, once it gets made, it's happy to float around, so to speak, and give off electrons where it's needed. And so this is a nice thing to have, because as you're running your physiology, you need electrons getting put into those pathways. And so NADPH is good for that. So all of that, what we just described, are the so-called photochemical or light reactions of photosynthesis in a nutshell. And then they're also showing on here a part two, what I call part two of photosynthesis, which is when the plant will take in carbon dioxide and make sugars from that. So that's called the Calvin cycle. And it's a cycle. It goes round and round and round and round all day long. And as that cycle is cooking along, what you end up getting is the production of sugars that the plant can use to build new material and so on. So chloroplasts were once free-living bacteria. So cyanobacteria, this is what they're doing. So plants are doing this, yes, but so are cyanobacteria. And I'm going to jump to this. So here you see a little bit more about the idea of an electron transport chain system. And again, don't get bogged down in the details, not in this class. In plant phys, we get into the details. But in this class, it's the idea of you have water being stripped of electrons. It's getting sent through this electron transport chain. So all the black arrows are showing the pathway. And then you can see the terminal electron acceptor right there, NADP+. And once it gets an electron, it gets turned into NADPH. And that's good. That's a major goal for the plant or the cyanobacteria. And then the Calvin cycle is being shown up here, taking in the CO2. So plants do this, cyanobacteria do this. And that's great because one of the byproducts is oxygen. But again, the plant's doesn't really care about oxygen. We do. We certainly do. But their goal is to get electrons sent into the system, into the electron transport chain. And in this case, cyanobacteria and plants, they'll use water as that source. But that's why you need a good dose of light energy, because water doesn't want to give up electrons. Questions on this idea with cyanobacteria and plants? Okay. So my hope is that some of you who've taken plant classes, this should sound fairly familiar, but if you've learned a little bit about this in intro biology, for example, or one of your chemistry classes, I wanted to talk about this first to kind of get your brain running. But now I want to show you that this isn't the only way to do photosynthesis when you talk about microbes. So now we have this idea that when you think about the sun and sunlight energy striking the earth, I said plants like to use red and blue light. when they do photosynthesis. Their chlorophyll pigments are adapted to absorb that really well. And that's great. But here what you're seeing is different pigments absorbing different wavelengths of light. So not all organisms that do photosynthesis will have the mixture of pigments like a plant does. They'll have different pigments that they use to absorb light energy. And what this is trying to show is on the bottom, you have, if you shine sunlight through a prism, a glass prism, it breaks into all those lovely different colors, the different wavelengths, red, orange, yellow, green, blue, indigo, violet. And so plants like to use the red and the blue. And anywhere you see a peak, that's their preferential wavelength, that their pigment is evolved to maximally absorb. And you can see they don't all match up. All these different pigments, they have different absorption peaks. So what that's saying is these microbes that are doing photosynthesis have different pigments, absorbing different wavelengths of light, but that's a good thing because then they can hang out together and not have to be competing for the sunlight. So those stromatolites we talked about, how we have matting going on where bacteria are hanging out in dense populations. You can imagine where there might be a bacteria, piled up on another one. And if they all want the same wavelength of light, the ones that are getting shaded out lose out. But what if they are like, yeah, you can have the red and blue light. I don't care. I'm going to have a different wavelength. And so you can imagine a situation where there might be microbes in very close physical environments, and yet they're not competing for that sunlight, the same wavelength of sunlight energy necessarily. And so it allows for populations of photosynthetic organisms to hang out in really close contact. It's okay because they're all kind of eating their own wavelengths of light. They're able to take a different wavelength and use that. And so here too, if you think about aquatic systems, different wavelengths will go to different depths of the water. And so some wavelengths of light don't make it past the upper layers very well. Others, like red quality light, tends to go a little deeper. And so this is also showing that you can have microbes kind of hanging out at different depths in a water system based on what pigments they have, based on how sunlight can go and penetrate into a water column. So your Winogradsky column, that's what's happening right now. You have enough water in that column where you've created a nice gradient and you have them in the windowsill. And so you're creating a situation, hopefully, where eventually you'll see this, these microbes setting up home in different areas in that column based on how far the sun is reaching down into that water column, what wavelengths are doing that, and the microbes that have the pigments that can take advantage of that. And so if you get lucky enough, you may actually see these colors show up. Even in the sediment, you'll see some of these nice colors showing up where you can have phototrophs, especially toward the top half of sediment. You can actually see them once they start growing enough. And then in your water column, you're probably going to get a lot of cyanobacteria and maybe even some algae. And in the past, we've had some protists show up too that are in there and they're eating a lot of the microbes. And so it's this nice closed system. It takes a little time though for it to show up so we can see it with the naked eye. And then here too, they're pointing to the idea they're trying to get across. It says the penetration of light and anaerobic condition. So when you think about oxygen availability in water, if you've taken intro ecology, they usually talk about that. Oxygen doesn't really diffuse that well in water. It's slow. And so eventually you'll get at certain depths, you'll get to the point where oxygen won't even be there. It'll be an anaerobic state. So lots of things are happening in your Winogradsky column, not just photosynthesis. There's all kinds of things. There's lithotrophic things going on and other things too, but let's keep focusing on photosynthesis. So this is Again, from a microbiology textbook, and so they're zooming in on cyanobacteria. But basically, this is showing pretty much what we just looked at in the previous slide when I was talking about how plants do it. You still have the same players pretty much in the electron transport chain process. One thing I do want you to key in on, I forgot to mention, let me go back here. One thing for this information on photosynthesis, I do want you to key in on how many photosystems are getting used. All right, so photosystem 1 and photosystem 2, plants use that as their part of the electron transport chain. Cyanobacteria do that as well. So that's what you're seeing in this next slide. So this is how they've mapped out how cyanobacteria do photosynthesis, and you can still see photosystem 2 is showing up here and photosystem 1. So we say they have 2 photosystems. But again, it's the same, this picture is just showing pretty much the same thing where you have an electron transport chain process. And then you have NADP plus as the terminal electron acceptor. And you got ATP being made. But some microbes only have one photosystem. So this is, and there's a lot of debate about what came first. You know, did a 2 photosystem mechanism come into play first or 1 photosystem? Right now, it seems that the debate is leaning toward the bacteria that can do just 1 photosystem is what evolved first, and then a 2 photosystem strategy came later. But here you're seeing this, this is an example of green sulfur bacteria. That's the group of bacteria, that's what they call them. But basically you're seeing this idea here where there's only 1 photosystem. It shows up better right here. Okay, so just photosystem 1. Now this is a busy image, but basically you're still seeing the basic idea of the use of light energy, striking pigments. In this case, they're not chlorophyll, they're bacterial chlorophyll, BCHL. That just means they're not, it's slightly different than chlorophyll. But it's the same idea. You are getting this electron transport chain process. Where is it? Let me erase. So here, this green sulfur bacteria is taking hydrogen sulfide and stripping electrons off of it. And the blue line is the electron flow. So if you take electrons off of hydrogen sulfide, will oxygen be a byproduct of that? Can you get oxygen from hydrogen sulfide? No. So that's another key thing. These green sulfur bacteria, when they do photosynthesis, they're not making oxygen. So they're anaerobic. But you still have this electron flow all the way to NADP plus as the terminal electron acceptor. And so you end up with NADPH, which is good. And the artist didn't put on here that you do still make ATP from this because you are getting protons pumped across the membrane. So when bacteria, cyanobacteria, plants, here you're seeing these green sulfur, they're still producing, it's not shown here very well, a proton gradient. And so you still get ATP production. Okay. And that's similar to what we saw with plants and cyanobacteria. They're producing a proton gradient through this electron transport process. So you can see protons showing up across a membrane and getting a proton gradient. The artist should have been a little bit more generous with how many protons are down here, but you get a proton gradient and then you get the protons diffusing through the ATPase to get you ATP. So that's happening again, even if you just have photosystem 1. So if that's all you got to work with, if that's all you evolved, you can still get a proton gradient. You can still get ATP. And then, of course, you get your NADPH, which is good too. So this is a twofer. You get ATP and NADPH with this proton gradient process. Well, proton gradient is helping with ATP, but the electron transport process, getting the final product of NADPH, that's really good for the cyanobacteria, the bacteria, the plant. They use that. They like to get electrons off of that. So photosystem 1 is using, in this case, they're showing hydrogen sulfide. That's not the only thing though. Other bacteria that only have photosystem 1, they can use other stuff besides hydrogen sulfide. And so there's a lot of options here. That's just one example that's being shown. Then there's some other bacteria that just use photosystem 2. That's the only option they have. They didn't evolve the ability to use photosystem 1 and photosystem 2. So they just have photosystem 2, which is being shown here. Now, these are a little bit disadvantaged. Photosystem 2, this one's weird. So when bacteria are using photosystem 2, a couple of weird things going on here compared to what we have just been talking about. If you look at, here's the starting point for electron flow, right there. P870, that's the pigment. Okay, so 870, that's the nanometers, that's the wavelength of light that this pigment is capable of absorbing. And you see the electron flow is in blue. So follow the blue line. Ignore this for right now. Follow the blue line. Is it a linear pathway? It's circular, cyclical, circular. So it goes, this electron gets released off the pigment and goes around and then back to the pigment and gets hit with light again. Electrons given off and it goes around and around and around and around. Okay. You see the circular flow there? So that is an interesting twist on photosynthesis. Instead of having a linear flow of electrons from, you know, an ultimate donor to an ultimate acceptor, it just keeps going round and round and round. But they still manage to create a situation where there's protons getting pumped across the membrane. So they still manage that. So they're still linking it to proton pumping. So that's good because that gets you your ATP. But you don't see, I'll cover this up. You don't see along the blue line here, you don't see NADP plus anywhere there, do you? There's no NADP plus. So these bacteria are at a disadvantage compared to all the others we've talked about. They're not directly making NADPH. So that's kind of a bummer. That's not as good of a situation because it's really good to have NADPH. If you can make that through this process, that's really good. They're not able to do that. They have to do something that's called reverse electron flow, which I'm not going to get into, but it's something that's kind of tied to this, but it's a separate process. And it uses energy. They have to expend some ATP to make their NADPH. So that's not so good. It'd be nice to just capitalize on this, put it as the terminal electron acceptor, like we saw in the previous situation with photosystem 1, the bacteria that do photosystem 1, they're still able to use this electron flow to make their NADPH. And that's a really good situation. Those bacteria that only have photosystem 2, they have to expend ATP in a separate process that gets them their NADPH. However, they still end up getting some NADPH. They still end up getting ATP. It's just a little bit more energy intensive for them. So, last I read on the debate, it seems like people are leaning toward photosystem 2. Bacteria that can only use photosystem 2 came first. Then some bacteria that could only use photosystem 1 came next. And then eventually sticking it all together using both photosystems and creating a little bit more efficient and elegant process came last. But that's all still up for debate. So again, when you're looking at this, don't get bogged down in the details. You can see a lot of similarity with this game of electron transport chain. And then with the photosynthesis, we're looking at, well, how many photosystems are involved? Because that will really dictate if you do have some nice NADPH getting made. And what kind of flow of electrons do you have? Is it a nice linear flow from a donor, ultimate donor to an ultimate acceptor, or do you have this weird circular pattern? So as the electron comes back to photosystem 2, that pigment gets hit with light again, and that excites the electron off again, and it just keeps zinging around and coming back with light, zinging around. And so you get this weird circular pattern of electron flow. But it works. They end up creating ATP from that, so that's good. Okay, so 2 photosystems. Maybe they just have photosystem 1, maybe they just have photosystem 2. All using light energy though, pigments to absorb that in order to get electrons flowing through some kind of electron transport chain. When you're studying this, I would encourage you to compare and contrast. How do plants and cyanobacteria do it? How do these other groups that only use photosystem 2 and 1, how are they doing it? What's similar? What's different? That's kind of what I want you thinking about, not the nitty gritty. Okay? Yes.
Will we have to remember, like, distinctly between purple bacteria, green bacteria, whether or not one started off with photosystem 2, one started off with photosystem 1? Okay.
And you can easily look that up, but I'm more concerned, do you understand like the basic theory of how are they generally doing photosynthesis? So you don't necessarily have to memorize purple bacteria to do photosystem 2 or like certain species within that. That's a group name. There's lots of different species. Other questions on photosynthesis? The last couple of slide sets are terminology that you've seen some of this before. So this slide set, what I've done here is when you look at microbes or really any living organism on the face of the earth, you look at the idea of how are they getting their carbon? We get our carbon through eating something that was once alive. But some microbes can take CO2. So how are they getting their carbon? How are they getting their energy? We're using energy from chemicals. We don't do light. And then electron sources. How are we stripping electrons? We use chemicals for that. But microbes can do a lot of different things compared to us. So these 2 bullet points, I've paired them together. So these 2 bullet points are just saying, how are they getting their carbon? Some microbes take in CO2 and make sugars off of that, similar to how plants do it. But some are like us, we're heterotrophs. They have to get access to some organic molecule near them for a carbon molecule. So I paired those 2 together. It's an either or. Either you're an autotroph or you're a heterotroph. Then these 2 are paired together. How do you get your energy? You either absorb sunlight. Unfortunately, we can't do that. I would love it if we could do that. I wouldn't have to make dinner every night. And so phototrophs versus chemotrophs, we're chemotrophs. So a lot of microbes are chemotrophs. They have to strip electrons off of some compound. Phototrophs can use light energy for their energy. And then these two are an either or situation. You're either a lithotroph or an organotroph. We're an organotroph. But some microbes can take electrons off of minerals, inorganics. So when I do need you to memorize these terms, please. And I highly recommend you memorize it like this, where you're doing it in pairs, either or, either or, either or. Because otherwise it gets very confusing, I think. But if you can pair them up like this. And then, of course, you can start taking these and grouping them. And so the next couple slides are getting at, okay, photo, litho, autotroph. So you're taking from those three pairs on the previous slide. So photo means you're using light energy. Litho means you're using inorganics, stripping electrons off of that. Autotroph means you can make your own sugars. And then some examples of those. And then you can have, sometimes they drop the chemo off, but organoheterotrophs, chemo organoheterotrophs, that's what we are. And that's what most pathogens are that like to attack us. They are adapted to that too. Fungi are that way. And so most bacteria fit into these two categories that have been studied and isolated and looked at. And then the next slide really quickly here is getting at some more bizarre cases, photoorganoheterotrophs. And these are some of the purple and green bacteria. So you tend to find these in nicely polluted lakes and streams because that's where a lot of organic material will be that they need. Although some can use CO2. And then chemolithotrophs, chemolithoautotrophs. They're stripping electrons off of inorganics and they're also making their own sugar. So a lot of microbes that are recycling nutrients for us and for planet Earth, they're chemolithoautotrophs. We'll learn more about all of these groups when we look more at the Winogradsky column and think more about that. And then this last is just showing it in table form if that helps you study. Okay, so spend some time. I know it's a lot of jargon, sorry, but please spend some time memorizing these and thinking about what they mean. All right, we got to shift over to lab.